In today's lecture, we are going to talk about photosynthesis, the only process among living things that can convert the energy of sunlight into the potential energy of the chemical bonds. In addition to that, photosynthesis serves as the source of oxygen, which is essential for the survival of many, if not all, organisms. What essentially happens during the process of photosynthesis is that the energy of light that comes from the sun energizes the electrons, and then the energy of these electrons is stored in the form of chemical bonds within the molecules of various sugars. The only organisms on Earth that are capable of performing photosynthesis are plants, algae, and cyanobacteria. These organisms are called photoautotrophs. So these photoautotrophs, the top line of images, are self-feeders. Photo refers to the source of energy, and auto refers to the source of carbon. Most other bacteria, as well as animals and fungi, are so-called heterotrophs, which means they consume organic compounds that were produced by autotrophs. They essentially rely on the organic compounds produced by photosynthetic organisms. However, there are some bacteria that can extract energy from inorganic compounds and can use inorganic carbon sources to produce sugars. We call these organisms chemoautotrophs. Such chemoautotrophs can be found very often at the bottom of the ocean around the volcanic vents. Photosynthesis consists of multiple steps and requires a supply of carbon dioxide, water, and the presence of very specific wavelengths of visible light. In the process, photosynthesis releases oxygen and generates a carbohydrate, glyceraldehyde 3-phosphate, which you may remember as a high-energy intermediate in the process of glycolysis that is produced during the energy investment phase of glycolysis. Glyceraldehyde 3-phosphate can be further converted into molecules of carbohydrates, such as glucose or fructose. The process of photosynthesis takes place in the leaves of plants, specifically in a middle layer of a leaf, so-called mesophyll. Carbon dioxide and oxygen enter and exit plant leaves via special openings called stomata. The singular form is stoma, which, in addition to providing a passage for carbon dioxide and oxygen, also regulate water balance. When stomata are closed, water cannot leave the leaf, and the plant saves the water. When stomata are open, water unfortunately can leave the plant, and it has to be regulated tightly. Usually, stomata are located on the bottom of the leaf to minimize the loss of water, and the cells that regulate the opening and closing of stomata are called guard cells. Within a mesophyll are photosynthetic, within the plant cells, there are photosynthetic organelles that you heard about before called chloroplasts, which, just like mitochondria, are the product of endosymbiosis. They have a double membrane envelope, just like mitochondria, and these chloroplasts were derived from bacteria billions of years ago in the process of endosymbiosis. Another evidence towards this is that chloroplasts have DNA and ribosomes, which look just like bacterial ribosomes. Within a chloroplast, you can find disc-shaped structures called thylakoids. These stacks of membranes contain chlorophyll, a pigment that absorbs light, and also there is an electron transport chain within the membrane of a thylakoid. Thylakoids organized in the stacks form a granum, and grana, multiple grana, are surrounded by the gel-like substance known as stroma. The summary of photosynthetic process can be shown here. 6 molecules of carbon dioxide interact with 6 molecules of water, producing a molecule of sugar, glucose, and 6 molecules of oxygen. All photosynthetic reactions can be divided into light-dependent and light-independent. During light-dependent reactions, shown here on the left and shown here on the left, energy from sunlight is converted into a chemical energy of ATP and NADPH. During light-independent reactions, chemical energy that was harvested in the previous step, light-dependent reactions, the chemical energy stored in the molecules of ATP and NADPH, facilitates the carbon fixation, assembly of organic sugar molecules from carbon dioxide. This set of light-independent reactions is called a Calvin or Calvin-Benson cycle. Essentially, the energy of light is stored in the electron carrier, such as NADPH, that is formed in light-dependent reactions. ATP as an energy carrier delivers the energy to light-independent reactions, to the Calvin cycle, where this energy is used to form chemical bonds of carbohydrate molecules and therefore store the energy there in the potential form. Photosynthesis depends on the sunlight, which is an electromagnetic radiation. Solar energy reaches the Earth in the form of waves, and shorter wavelengths, wavelength is the distance between two peaks of two adjacent waves, two crests. Shorter wavelengths have a greater energy and vice versa. Electromagnetic radiation with the shortest waves or highest frequencies, such as X-ray and UV light, have such an enormous amount of energy that they can actually harm biological objects. Here in the illustration below, you can see that the visible light actually represents a very small part of the electromagnetic spectrum. Pigments that participate in the process of photosynthesis can absorb very specific wavelengths of light, somewhere between 700 and 400 nanometers. If the energy of that particular wavelength is sufficient, it will raise an electron from its ground state to the excited state. In other words, the electron will go from the lower orbital to a higher orbital. The radiation that is capable to do this, the radiation that is capable to excite electrons is called photosynthetically active radiation. Each pigment that participates in the photosynthetic processes absorbs very specific wavelength. And all the wavelengths that cannot be absorbed are either reflected or transmitted. Two major classes of photosynthetic pigments found in biological organisms are chlorophylls and carotenoids. Chlorophylls are comprised of 5 different classes. Chlorophylls A and B are found in higher plants. Chlorophyll C and D are found in algae, while bacterial chlorophyll is found in prokaryotes such as cyanobacteria. On the other hand, carotenoids represent an enormous variety and are commonly found in fruits and vegetables such as lycopene, found in tomato, zeaxanthin, found in corn, and beta-carotene, that is found in oranges and carrots. The main role of carotenoids isn't as much to carry out photosynthetic reactions, but rather absorb and dispose excess light energy, protecting chlorophyll from the damage. Each type of pigments, from carotenoids to chlorophylls, can be characterized by a specific absorption spectrum. For instance, in this image, you can see that chlorophyll A, shown here as the solid line, has the absorption peaks in a blue and deep red parts of the visible spectrum, which explains why chlorophyll A is green. Chlorophyll B has the peaks in a blue and yellow spectrum, which adds which explains why chlorophyll B is also green. On the other hand, carotenoids have the peak absorption in the blue closer to the green part of the spectrum, and they do not absorb light in the yellow and red part of the spectrum, which explains their yellow, red, and orange color. Usually plants and other photosynthetic organisms contain a mixture of pigments which allows them to use a wider range of wavelengths. Now we're going to talk about light-dependent reactions in the process of light absorption, first of all. Light-dependent reactions depend on 2 photosystems, photosystem 2 and photosystem 1, which convert the energy of light into chemical energy stored temporarily in the molecules of ATP and NADPH. Photosystems 2 and 1 consist of multiple proteins, and they share a lot of similarities. Both photosystems contain so-called antenna proteins, with the pigment molecules bound to proteins that form light harvesting complex. The role of the light harvesting complex is to pass the energy of light to so-called reaction center. So here you can see light harvesting complexes on both sides of the reaction center. When a photon of light is absorbed, it excites the molecule of a pigment, and then the energy is transferred from one chlorophyll molecule to another until it reaches the special pair of chlorophyll molecules in the reaction center complex. When this energy is transferred to the reaction center in a photosystem 2, the chlorophylls in the reaction center are oxidized. In other words, they lose electrons, committing so-called photoact. The cytochrome complex within the electron transport chain of photosystem 2 transfers the electrons from reaction center to plastoquinone. Electrons are transferred from the reaction center to plastoquinone, and then cytochrome complex transfers the electrons from plastoquinone to plastocyanin. Plastocyanin eventually directs those electrons to the photosystem 1. The transfer of electrons from plastoquinones through cytochrome complex to plastocyanin supplies the energy for the transfer, the pumping of protons across the thylakoid membrane, where protons move within the thylakoid space. Eventually, the electrons are delivered to the reaction center of the photosystem 2. In the meantime, one must replace the electron that the reaction center in photosystem 2 lost, and the replacement of this electron occurs when water is being oxidized. More specifically, oxygen atoms in the molecule of water are oxidized. This oxidation results in the production of hydrogen ions and molecular oxygen, and the electrons reduce the reaction center of the photosystem 2. You can see this reaction right here. Approximately 10% of the oxygen that is generated during the process are used by the mitochondrial plants, while the rest of it is available for use by other aerobic organisms. This looks very similar to what we've just learned about cellular respiration. Essentially, the energy of electrons in electron transport chain is used to pump hydrogen ions from the stroma across the thylakoid membrane into the thylakoid space or thylakoid lumen. These hydrogens that accumulate in the thylakoid lumen will be used to synthesize ATP. Now, let's go back to electrons. If you look at the previous image, the image right here, the electrons that were energized in a photosystem 2 have now arrived to the photosystem 1, and they have lost their energy before the arrival. Therefore, these electrons must be re-energized in a photosystem 1. Photosystem 1 absorbs another photon, and the energy of this photon is relayed to the reaction center in photosystem 1. Photosystem 1 is oxidized again and sends the high energy electron to the molecule of NADP+, an electron carrier characteristic for this process. The addition of these electrons to NADP+ results in the formation of NADPH, the reduced form of the electron carrier. To summarize the steps of light-dependent reactions, we can say the following. Photosystem 2 captures the light energy, creating the proton gradient, which will be used to produce ATP. Photosystem 1 captures light energy, which is used to energize electron to reduce NADP+ to NADPH. And the amount of NADPH produced is roughly equal to the amount of ATP generated. Now we've mentioned that the proton gradient is used to produce ATP, but how? This is the process you should be familiar with at this point. It is chemiosmosis. Elevated concentration of hydrogen ions within the thylakoid lumen essentially creates a gradient of concentrations across the thylakoid membrane. The diffusion, facilitated diffusion of hydrogen ions through the ATP synthase, the same process that we observed in the electron transport chain during cellular respiration, supplies energy for the ATP synthesis from ADP and inorganic phosphate. Now, at this point, we have harnessed light energy in the form of highly energetic electrons carried by the reduced form of the electron carrier, NADPH, and in the form of chemical bonds within the molecule of ATP. Now, this energy that was harnessed in light-dependent reactions is going to be used in Calvin-Benson cycle to produce organic molecules. When carbon dioxide enters the leaves through the stomata, it eventually reaches mesophyll cells, where it diffuses into the stroma of chloroplasts and participates in the Calvin-Benson cycle. So before we transition to Calvin-Benson cycle, we need to mention something. You have to remember that light-dependent reactions occur in the membrane of the thylakoids, and the thylakoid space is also involved, while light-independent reactions are confined to the stroma surrounding grana within the chloroplast. Now, I apologize for that mishap in the title here. Obviously, it is not bulk transport. It's photosynthesis. But the whole Calvin-Benson cycle can be roughly divided into 3 different steps. The first step is carbon fixation. The enzyme called rubisco, or ribulose 1,5-biphosphate carboxylase, facilitates the reaction between carbon dioxide and the molecule of ribulose biphosphate. When one molecule of carbon dioxide, which contains 1 carbon, interacts with 1 molecule of ribulose biphosphate, which contains 5 carbons, this process generates 2 molecules of 3-phosphoglyceric acid. So the term carbon fixation refers to the fact that carbon dioxide is fixed from its inorganic form into organic molecules. Carbon, oxidized carbon found in CO2, is now a carbon found in phosphoglycerate. The next step of Calvin-Benson cycle is reduction. 6 molecules of 3-phosphoglycerate and are converted into 6 molecules of glyceraldehyde 3-phosphate using 6 ATP and 6 NADPH. One may ask, wait a minute, we just talked about 2 molecules of phosphoglycerate, and suddenly there are 6. Well, consider this. Imagine that we triple all the numbers from that equation. Instead of 1 CO2, we're going to talk about 3. instead of 1 ribulose biphosphate, we're going to talk about 3. So we have 15 carbons in 3 molecules of ribulose biphosphate, 3 carbons in 3 molecules of CO2, which adds up to 18, and we have 18 carbons in 6 molecules of phosphoglycerate. The reduction step ultimately results in the formation of 6 molecules of glyceraldehyde 3-phosphate, which also contain 18 atoms of carbon. During that process, ATP supplies the energy for the reduction, while NADPH serves as the electron donor, being oxidized to its NADP plus form, which will be recycled and used again in the light-dependent reactions, just like adenosine diphosphate and inorganic phosphate. The third step is the regeneration. 1 molecule of glyceraldehyde 3-phosphate leaves the Calvin cycle. This molecule will be used to form other organic compounds, such as glucose. If you think about this, when we talk about single turn of Calvin-Benson cycle, the single turn implies that 1 molecule of CO2 reacts with 1 molecule of RUBP, resulting eventually in 2 molecules of G3P. Not immediately, but eventually. Therefore, in order to export 1 molecule of G3P, one needs to turn that cycle 3 times. So 1 CO2, 2 CO2s, 3 CO2s, 3 carbons will be converted into 1 3-carbon intermediate used for the synthesis of glucose and other organic compounds. Now, it's important to understand that while 1 molecule of G3P it leaves the cycle, 5 other G3P molecules remain in the cycle. 5 G3P molecules here contain total of 15 carbons, just like 3 molecules of RUBP. These 5 molecules of G3P are used to regenerate ribulose biphosphate, and therefore to continue the cycle again and again. So the energy flow in the nature can be represented by 2 core processes, photosynthesis and cellular respiration. Photosynthesis, shown here on the left, absorbs the energy of light and forms chemical bonds, carbohydrates, in chloroplasts. Cellular respiration on the right uses the energy stored in those chemical bonds, releases it using oxygen to metabolize those carbohydrates. Both processes use an electron transport chain, but chemically they are essentially opposite. However, both processes are essential to fuel other reactions in the cell. In our next lecture, we're going to talk about DNA structure, DNA function, and DNA replication.